How about now?
Perfect.
Okay. Let me.
And the E floor is yours.
Okay. Thank you so much. All right. Very good. Okay. So now, do you also see the pointer?
Yes.
Perfect. Thank you so much. Yeah, so today I would like to share some of our experiences related to clinical translation and discuss with you where and how biofabrication fits into this big picture. So over the past 30 years, we have followed this tissue engineering strategy, which involves the use of cells, biomaterials, and biological factors to build tissues for clinical applications. We sometimes use cells alone or scaffolds alone, or many times if the extent of damage is greater, then we would use cells and scaffolds to engineer tissues for reconstructive procedures. And it really depends on what the application is and what tissue types you're trying to repair and provide treatment to patients. So using this strategy, how do we do it? So this is an example of an approach that we have followed in real patients, and we always start with, from taking a small tissue biopsy from a patient, and we would, take the tissue biopsy, isolate cells of interest, and then expand it in large quantities in vitro, and then place them onto a supporting matrix, which usually consists of biodegradable polymer type of scaffolds. And then we would precondition it in a bioreactor system of some sort, and then that entire construct, when they are matured and preconditioned, gets transferred into the OR and use it for implantation and repair the tissue damage. So using this approach, we have brought a number of tissue applications to patients. And bladder was actually the first internal organ that we have developed and implanted in patients to treat pediatric patients with congenital anomaly in 7 patients. And we have successfully followed the patients for over a long period of time. Similarly, we have also developed and treated patients with urethral stricture, which is the obstruction of urethra, which a patient cannot void due to traumatic injuries. And these patients were also treated using this approach and have followed for over a long time. Another application that we had developed was to engineer vaginal tissue in patients who were born without vaginal canal. And this is related to congenital anomaly involving vaginal aplasia. And these patients we had built and restored normal tissue, and these patients were followed, and during that follow-up, all these patients grew up and got married and have a happy life. So, this is very exciting because, unlike bladder or urethra, where you're trying to treat patients with tissue damage, we're able to create a new organ for patients so that that patients can live a happy life. So as you can see, we have developed many tissue engineered products or tissues and had initial successes in building them and providing the patients with the tissues that they need in order to live a normal and better life. However, there are challenges, and those challenges do exist in developing when you're trying to develop and translating a more complex tissue systems that require either systemic function or coordinated function. And these are some of the developments that we're currently doing at our institute. Now, this corpora tissue is a highly vascularized solid organ, which we're approved to do clinical trial, and we're just initiating the first patients in the very near future. And uterus is, the most recent development that we were able to create uterine tissue, of course, in an animal model, and we showed that this engineered tissue supports live birth, as you can see here in a rabbit model. So these are very exciting developments. Of course, we cannot bring these technologies immediately to patients, but we're working very hard to further develop and refine it, and make sure that they're safe for patients. So these are all the developments that we're doing. But however, getting back to the challenge issue, there are certain challenges that we have to address in order to bring any technologies to patients. So one challenge that I would like to bring your attention is the engineering challenge. And all the applications that I have shown you in previous slides were done manually. We fabricate the tissue, the scaffold manually. We culture cells manually. So everything is done manually. However, as you know, the body is a very complex and delicate organ and tissues, which requires a very refined approach. We are unable to replicate the natural vascular network that we have in our body, or the organ and tissue complexity of each specific organs and tissues. And that is problematic when we're trying to build such tissues. So that's when bioprinting got our attention, because we were looking for ways to find a better way to develop tissues for patients. And bioprinting back then, this was 15 or 16 years ago or more, we're trying to look for opportunities and technologies that would allow us to build better tissue. And bioprinting technology allows us to deliver many of the building, tissue elements that are required to develop into tissue, which includes cells and biomaterials and a lot of biological factors that you could actually deliver them in a three-dimensional and spatial manner to build a scaffold that could be implantable. So bioprinting technology, in summary, is a fascinating tool, an able tool to build implantable tissues and organs, which allows us to scale up into a clinically relevant size with precision and reproducibility, which we cannot perform with our own hands. So this has been a technology that we really sought after. So in early days, printing was just beginning. So we, recruited Tao Zhu from Clemson, from Tom Boland's lab, and built an inkjet printer with a modified inkjet printer that would have an elevator platform acting as a z-axis, and so that platform would depress one cell layer deep as you print over and over again. And using this approach, we were able to demonstrate that yes, printing technology used for building tissues. As you can see here, we were able to print, with cells, a beating heart. If you deliver stem cells that are differentiated into bone, they actually form bone. Likewise, blood vessels. But what excited us was to demonstrate that we can deliver multiple cell types into a small confined space in an organized fashion, as you see here. And those, when they are implanted after printing, are able to mature and transform into tissues with those specific cell functions and tissue functions. So that's very exciting. And after that, we realized that inkjet printer has a lot of limitations. So we started building different printers for different tissues, and this is an example of a skin printer we have designed and built and have tested in a porcine model, as you see here. And this is another iteration of the skin printer that we're trying to further develop so that it can be used in the clinic. Another type of printer that we built was integrated tissue and organ printing system, which allows us to deliver and generate a 3D freeform shaped constructs for different types of applications with precision and reproducibility. Nowadays, this was back in the old days, over 12, 13 years ago. But nowadays, there are many sophisticated printers that are available for many different applications and research purposes. So you should consider yourself lucky. So it took us so many years to build this unit, and but we have tested that we can print a very fine with precise tissues organoid structures that we could use it for many different purposes. But using this printer, we are able to build tissues that can be implanted in an animal model. And the great thing about bioprinter is that you're able to design and place whatever tissue element that you want to place in a precise location. As you can see here, you can design it, you can shape it, and you can place cells in different orientations, as you see here, and you're still able to generate a tissue that can be surgically implanted in animal model where the tissue construct would further mature into functional tissue, as you see here. We have tested whether bioprinter is beneficial in developing tissue constructs, and we have demonstrated in several applications that if you place cells and other materials in an organized fashion, you would further better mature and get enhanced achieve enhanced enhanced function, as you see here. So using this printer, we have been able to develop many different tissue and organ types, ranging from soft tissue to hard tissue and simple tissue to complex tissues, as you see here. And most of these developments have been tested in preclinical setting and demonstrated that tissues can be formed in vivo. So, what we're currently focusing on is, developing composite tissues using printer because, many of the tissues that are required for repair in vivo has, composite tissues in nature. Tissues with different tissue properties or different cell types as well as different functions. So we're really focusing on these composite tissues like bone cartilage interfaces and muscle tendon interfaces, as you see here. So are we going with the bioprinting technology? We do envision using this in the clinic where we could use their medical data to generate, tissue constructs that are printed from the bioprinter, which can be delivered to the operating room so that the surgeon can use the natural tissue for reconstruction. That's where we do see this technology going. And I often get asked this question. You guys are doing printing many different tissues. Why is it not in the clinic yet? Or when will it be available? And that's a very valid question. However, we all know that it's the although the technology is has proven and is there, we do need further development on many of the different components. When people think of bioprinting, well, you put cells and biomaterials and just print. What's a big deal? But we all know that there's a a very sophisticated, science behind bioprinting. That's why we're, working hard, to today. If you, segment the bioprinting technology, you could, separate it from hardware, software, and, bioinks, that would work in concert with each other in order to produce a tissue construct that can be implantable. And, we know that the current hardware that are available are not, there yet for clinical use readily. Software too. The most of the printers that we utilize in the lab is very slow, and there are many improvements software-wise in order to treat patients clinically. And there's a lot of activity going on in developing bioinks because there are many biomaterials, but those are not formulated or set up to use as bioinks. So many of you have been working on developing a bioink that can be reliable and safely used in the in the lab as well as in the clinic in the future. But there are other considerations, regulatory pathway. Because that we're able to print and implant in a preclinical model, that does mean that it can be used in patients. So there's a lot of regulatory pathways that we have to go through in order to demonstrate that the product that you're delivering to patients is safe and biocompatible. and then we have to figure out a way to distribute this, and we can only do this commercially. And so developing a strategy to distribute is another challenge that we have to figure out. And so, in addition to that, there are other, scientific and technological challenges. Just because we're able to, develop a tissue construct that is, let's say, identical or similar to the tissue that we have in the body, how are we going to transfer it into our body? Obviously, because these constructs have cells, they need to be vascularized, and your implant has to be innervated in order to function, as well as many other challenges and limitations we currently have. We do need to identify them and overcome those challenges. So I would like to touch a few. That we're currently being very focused on developing. Obviously, vascularization is a challenge that everybody's aware of for many decades, and so for vascularization, many investigators around the world have tried to introduce many different ways to accelerate the angiogenesis or neovascularization. So people have used biological approach like using vascular endothelial growth factors or endothelial cells to promote vascularization. Other approaches also is trying to embed oxygen generating particles to provide oxygen to surviving cells until the host vascularization is established. So that's one potential approach that could solve the problem. And yet another approach that was introduced several years back was to downregulate the cells that you put into the body by decreasing their cellular metabolism so that they would require less energy. So these are some of the approaches that investigators have tried in order to overcome the vascularization issues. Other physical approach is, maybe when we develop a tissue construct, can we pre-vascularize them so that when we implant, the host vascularization would naturally connect to the pre-existing pre-vascularized channels. So when you implant a tissue construct, it takes a very long time because your body makes new blood vessels and the infiltrating new vessels, kind of infiltrate in from the periphery. And so it does take time for the host vessels to, generate a tissue, vascular tissue or vasculature into the tissue construct, which takes weeks. And it would be impossible for the implanted cells to survive during several weeks, depending on how big your construct is. So so the strategy is to pre-vascularize the construct so that as you develop your tissue construct, the vasculature or vascular network will be already existent, so that the host vessel would have to come in from the periphery and readily connect into the existing channels. So by doing so, several approaches or means have been introduced, including bioprinting or patterning or casting method, which all try to achieve the same thing. Pre-vascularize so that the body would find its way and readily connect so that the cells that you implant will be viable and mature into tissues. Otherwise, if the cells necrose, then what good is it if and all the effort that you put in to develop a tissue construct. Those are these are some of the approaches and attempts that have been made, and in our institute, we have tried, all of those approaches to overcome the vascular challenge. And this is an example of the, biomimetic vascular scaffold using the casting method, where you would take a native tissue or organ and then infuse it with a casting material, casting polymer, and fill all the vascular channels in the kidney, and then you digest the tissue, leaving only the cast, and then a polymer, scaffold polymer is used to coat it, and then after it's coated over the cast material is then removed, creating a hollow vascular scaffold with native, vasculature identical to native vessels, as you see here. And you can use that as a scaffold, seeding different cell types, endothelial cells on the inner side, as well as, other, cells of interest. So this method is one of the promising way to vascularize a scaffold. And this shows a cast, and it does preserve a very, microarchitecture of the kidney in this case, while preserving the vascular network that can be perfusable. And to that you can seed cells on it and implant it. And when you implant it in vivo, these vascular channels are able to integrate into the host blood vessel to provide, vasculature or blood to the implanted tissue. So, this is, one exciting development that may allow us to bring, clinical tissues faster. And many groups, around the world are trying to bioprint vasculature in, in order to overcome and address this challenge. Other technologies that we have been working on is to develop bioreactors. And as you all know, that bioreactors, are used to precondition the tissue construct that you develop, whether you do it manually or with bioprinting. So it does precondition and make sure that the tissue can survive and can do well in the given environment in vivo. And also it helps cells to mature into tissue-like structures in vitro. And we're actually currently using like bioreactors for bladder and urethra and corpora in the clinical trials as well. So because you're dealing with different types of tissues and organs, the bioreactors has to be different and very specific to the target organs that you're trying to build. So, yet another, development that we're currently, exploring by the team at, our institute is to develop a universal media. So many of you have, are working with different cell types and multiple cell types. But the problem is that each cell type has different requirements, nutrient requirements. So we do use different medium for different cell types, and that gets very cumbersome when you're trying to build tissues that has multiple cells, cell types. Like kidneys, it's an extreme example, but kidneys have, almost 30 different cell types. But in those cases, what media will be beneficial and will be right for different target cells. So our team at our institute are trying to develop a universal media that can be used for all cell types. So that this is uh, you know an exciting development which would eliminate the variability and inconsistency between different medias that you use for actual applications. So, these are some of the developments that are required and would be helpful in order to accelerate clinical translation. As clinical translational pathway is a very long pathway, which of course starts with science, research and discovery phase, but there are other paths that you have to go through and in order to finally deliver your tissue product to patients. Of course, for each phase there are many challenges and many opportunities to further develop and improve your developments. We talked about many of the challenges in scientific, but there are you do have many different challenges in process development and manufacturing the product or scaffolds or constructs for clinical use, and you do need to have a standard procedures, as well as many of the different you need to be able to transfer and be able to scale up to the actual human applications. Obviously, regulatory component is also important, and it does take a long time to satisfy the regulatory agencies because you do need to demonstrate that the construct that you're building is safe and biocompatible, and it's not toxic, and it's not going to cause tumors. So this is a very long, lengthy process. That costs a lot. And then, of course, I mentioned about commercialization. And there are many different factors that you have to decide, and how you want to disseminate this great technology that you're developing. So overall, it just takes a very long time to develop a product, tissue product that can benefit patients. Many of you are engaged in this process, but if you're in the beginning or are much farther away, there are some practical aspects or considerations that you have to consider. So we have developed a successful product in the lab. That doesn't mean that it will be successful in transferring the technology and we have to, we all start our research because of the clinical needs, but then, clinical needs and market needs are very different. So you may want to look into, how many people will really need this technology before you even start. And scientific and clinical outcome doesn't mean that you're going to be successful in the market. And even if we bring a technology to patients, through phase 1 clinical trial, that does not mean that you can automatically advance to phase 2 clinical trial, because it all depends on the outcome and cost and, market competitiveness. So you spend, 10 years, 20 years in developing a technology that you're hoping to, help patients, but then a better technology comes out much earlier, then, your competitiveness disappears. So, all these are very important practical considerations that you would have to, consider when you're trying to develop a bioprinted construct, for, whatever the application is. So, in order to minimize the risk, you may need to engage your, regulatory agency, your clinicians, and industry partners at the early stage of, scientific discovery. That's going to minimize and, save a lot of time and energy and costs in developing your products. But we all know that, and we are all hopeful that, this technology will be, useful in translating many different tissues and organ types. And of course, we're not going to build a kidney or liver tomorrow, but, like anything else, you do start with a more, simple and structural tissues first, and moving into a more complex systems over time. So, in conclusion, I think bioprinting technology or biofabrication is a fascinating field. That is able to generate clinically relevant sized tissue constructs with the precision that you cannot achieve with your hands. And of course, there are many scientific and technological challenges which we'll have to work through and address in order to develop translatable tissue systems. And of course, we do need to develop new and innovative technologies, so that we can get to our destination faster. So overall, bioprinting technology is an excellent tool that could accelerate translation, and we are, at the right time, when this, technology is prospering, and we're very hopeful that our approach will change the way the medicine is practiced in the future. So lastly, I would like to thank our institute members, and we're actively working on the different tissues, and we currently have 15 ongoing or planned clinical trials at the institute, and this could not be possible without the support from our sponsors who share the same vision as we do, which is to improve patients' lives by developing regenerate, new therapies and innovative tools, for patients. And this is the last slide. Please do save the date. We're going to have our annual meeting this year in Wollongong, but due to the COVID-19, this has been postponed to next year. So please mark your calendar, and we would like you to be there and maybe meet in meet face to face. And if you are not a member of our society, please join our society so that we can advance the new technology, the fascinating technology forward together. Thank you.